I think we're going to start now. Welcome everyone to our webinar for the Global Frailty Network. My name is Gustavo Duque. I'm the Kaufman Chair in Geriatric Medicine at McGill University and Director of the Center of Excellence for Sustainable Health for Seniors, also at McGill University, Montreal, Canada. So before we moved into the presentations for today, I just want to make a couple of housekeeping announcements. The first one is just for people who have not been in these webinars before, the Global Frailty Network is an international consortium that was created with the objective of promoting science in the frailty subject all over the world and from all disciplines, from basic sciences to clinical to community practice. So you are all invited to join our Frailty Network. Our website is under construction, but we will be available soon. But in the meantime, if you are interested in joining us, please feel free to contact through this webinar links. Also a reminder that these sessions will be recorded, but at the same time, we will post it at several websites that will be shared with you later on. And for the purposes of this session, questions and answers will be responded at the end. We want to encourage a lot of interactions. So what we're going to have today is three relatively short lectures, followed by lots of time for questions and answers and discussion. So if you have any comments or any questions during the sessions, please feel free to write them in the questions and answer function of Zoom. And finally, just a friendly reminder about the International Conference on Frailty Sarcopenia Research, which will be in Toulouse this year from March 22nd to 24th. And our Global Frailty Network, we have a strong presence at the meeting, not only by a symposium that was organized in collaboration with other consortia, but also through our place to interact with the attendees. So with no more introduction and housekeeping, I would like to introduce now our speakers. Very distinguished panel of speakers today. I'm really lucky to be chairing the session. Today we're going to focus on mechanisms, biological mechanisms of frailty from different approaches and perspectives. So we will have three lecturers from very well known around the world from three different organizations, institutions. The first speaker will be Dr. Luigi Ferrucci, who is a geriatrician and an epidemiologist who conducts research on the causal pathways leading to progressive physical and cognitive decline in older persons. He has made major contributions in the design of many epidemiological studies conducted in the United States and in IKEA study of centenarians in Sardinia and the Women's Health and Aging study. He was also the principal investigator of the INCHIANTI study, a longitudinal study conducted in the Chianti in September 2002. And he became the chief of the longitudinal studies section at the NIA. From 2002 to 2014, he was the director of the Baltimore Longitudinal Study on Aging, and at this moment, Dr. Ferrucci is the scientific director of NIA, which a position that he assumed in May 2011. Our second speaker is Dr. Susan Howlett. Susan is a professor of pharmacology in the Faculty of Medicine at Dalhousie University. She also has a cross appointment in the Department of Medicine, specifically in the Division of Geriatric Medicine. Dr. Howlett grew up in Montreal, where she obtained a Bachelor in Neuroscience from Concordia University, and she then completed a Master's and a PhD in Experimental Medicine at Memorial University in St. John's, Newfoundland. Her work at Memorial focused on skeletal muscle physiology and pharmacology, with an interest in muscle remodeling in an animal model of muscular dystrophy. At Dalhousie University, she's now exploring the impact of age and frailty on cardiac structure and function, as well as the role of sex steroid hormones in regulating the heart. And last but not least, Dr. Christiaan Leeuwenburgh received his PhD from the University of Illinois, where his doctoral work focused on the regulation of glutathione homeostasis during chronic glutathione deficiencies and or supplementation. Dr. Leeuwenburgh completed postdoctoral studies in internal medicine at the Division of Geriatrics and Gerontology and Division of Arteriosclerosis, Nutrition and Lipid Research at Washington University School of Medicine in St. Louis. He, at this moment, he is, in 2005, he joined the newly created Department of Aging and Geriatric Medicine, College of Medicine, and Institute of Aging at the University of Florida, where he functioned as the chief of the Division of Biology of Aging for the department. He has joined faculty appointments at the departments of Anatomy and Cell Biology, Biochemistry, and Molecular Biology, and is a member of the department's doctoral research faculty of the College of Medicine. His major research focus is to understand the molecular mechanisms of iron homeostasis, oxidative stress, and ferroptosis with age. So with, I think we have, if I read the whole bio, I will spend probably 3 hours. So with no more preamble, I would like to invite Luigi to share with us his slides. Thank you very much.
Let me unmute myself. That's better. So thank you for the nice introduction, Gustavo. And it's really a pleasure to be talking in this meeting. After the invitation, I was looking at put together a few slides to do a short introduction, a short talk. I couldn't really resist of making an epidemiological demographic introduction, which is absolutely important here. So the next slide, Christiaan, I am not seeing the next slide, but are you seeing the next slide?
So Luigi, I think you have to show the whole full screen.
Okay, I think that Christiaan is showing the slide for me.
Is that better?
Yes, perfect.
Yeah, could you go to slideshow? Yeah.
I wanted to show this to, give you a size of what is happening at the demographic level. And maybe this audience doesn't need that, but to look at, what is the structure of the population and they're going through. 2019 was yesterday, 2050 will be tomorrow, and yet, the yellow part of our population, the old part of the population will be expanding tremendously, more than double, in this short period of time. And the next slide tell you what the consequence is. Unfortunately, what we have is that as the average life expectancy, the expected year with disability also increase. And that is like a taxation that we get from every accomplishment, this tremendous goal of increasing the human life expectancy. And then the next one, I think that is what worries to me even more, is that the years lived with disability is correlated with health expectancy, with that expenditure. So in a way, the more money we spend in healthcare, and the more the year of disability are expanding, even percentually, that is true. And this is because what we're doing is prolonging the life of people that already developing disease, and we are impacting very little on the prevention that will delay the beginning and onset of disease. I think that if we look at the scenario of morbidity in the next slide, what's really happening, it's very clear, that we have experienced in our aging of the population has been due to the decline in heart disease and cardiovascular mortality. There has been a slight decline in the mortality for cancer, although the mortality for cancer in recent years declined even faster than that, especially with the application of the CAR-T. And there has been a decline in the stroke. but other diseases such as chronic respiratory disease, Alzheimer's disease, and the typical disease that are associated with complex multimorbidity, disability, and frailty tend to remain stable in the population. In fact, if you look at multimorbidity as a cause of death, the multimorbidity as a cause of death in many, many countries in the world has been expanding. The next slide. And the problem it's really outlined here. You know, the expansion, percentually, the expansion in the health, in the portion of life that characterized by disease and disability has increased more than the increase in life expectancy. And of course, you know, it's very easy to say that in absolute term, this may not be true, but somebody told me that, you know, the year that you live with health, enjoying life go very fast, while the year that your disability and frailty, it's much, much slower in the perception of the individual. The problem that we're dealing with is illustrated in my next slide. And the issue is that we're discovering that most of the disease occur in clusters. So that if you look at life expectancy, disease-free life expectancy, you know, the first disease that occur break this state of good health. And then many, many years ago, you know, the epidemiologists used to say, what's going to happen to the population if we could eliminate cardiovascular disease? And I think that was the wrong question, because in reality, the emergence of the first disease is correlated to a global susceptibility to disease that people are developing. And so that if you look at free life expectancy, that is in some individuals much longer, but yet after that period, the same cluster of disease occur. And in some individual, there is an early breaking of that disease-free life expectancy, and yet the same cluster of disease occur anyway. In a way, we can say that the first disease is increased significantly the probability that you will develop a second disease. So that is the epidemiological correlate of this phenomenon. And what we can do about that, I think that you know, the geroscience paradigm in the next slide is approaching things in a different way. Instead of preventing the first disease, which will be followed by the second one, because the diseases are developing in cluster, we can try to approach the susceptibility at the root. And the susceptibility at the root, according to our hypothesis, is due to the biology of aging. It's due to the fact that some individuals tend to age faster than others. And so by addressing aging, we will be able to slow down aging and reduce the burden of multiple disease instead of having to prevent or treat each specific disease at a time. So what is the underneath concept about this idea of the biology of aging? And here I've been trying to summarize, you know, the way we conceptualize it. And so at least in my group, and the way we think about it when we, you know, design our studies. So at the beginning of life, there is a very strong resilience. But resilience means that the entropic damage that occur at the level of molecule and cell is counteracted by a number of resilience strategies that have been developed over time and that have been selected evolutionarily. The problem is that these entropic stress also occur at the level of the resilience strategy, and so that as resilience is reduced, the accumulation of damage increase and increase until, you know, it's transformed into a functional problem. Now, frailty, at least from the perspective of the geroscience hypothesis, is a condition when there is no longer resiliency, and where even the stress of normal life is too much to be dealt with from the individual and lead to the rapid accumulation of damage with rapid mortality. And we know from our study and from other people's study that there are many ways that we can increase the resiliency, and there is also other stressor that can accelerate the accumulation of damage. I'm not going to go into detail because otherwise it will be too long. So how we transform this into conceptually in a way that we can translate in our studies. And I think that it's really important that we think this way as 3 levels that are highly and causally interconnected. The first one, that is what we really want, is to prevent the decline of mobility and the decline of cognition, because these are really what affect the quality of life of people. But we know that in order to do that, we need to look at what are the phenotypic manifestations of aging that eventually translate into this functional decline. And again, we need to understand what are the underlying biological mechanisms that are causing this accumulation of damage at the phenotypic level. And as you know, people have been trying to understand which one are, the basic mechanism of aging. I don't think that this discussion is finished, but if you go to the next slide, I put in the first line those that everybody has recognized from Lopez-Otting and later. What are the mechanisms that have done that? And so, tissue regeneration, epigenetic adaptation, mitochondria, I will not go into the details, but to tell you that the dysfunction of this network of resilience strategies is translated into aging phenotype. And schematically, we can think about the aging phenotype as something that is tangible and visible. Change in body composition, the reduction of energy, the fact that our homeostatic mechanisms tend to be not so functional as before, and then neurogeneration, neuroplasticity. And what is important here is that if you think about these 4 domains, there really are affecting what we call the geriatric syndrome. Think about incontinence. Incontinence really is a change in body composition that is joined with neurogeneration and neuroplasticity. Body composition is what is really causing sarcopenia, and then pain is what is really affecting functionally, probably the most important cause of mobility loss in the older population, and so on. So it's important that we think in terms of biological damage, phenotypic manifestation, and then functional effect, such as the geriatric syndrome. And one last word. There is a time gap between these 3 events. The biological manifestation occur early, and only after resilience is consumed, they create a problem at the phenotypic level. And again, the phenotypic manifestation only late cause a functional problem. This is important because that means that we can address molecular and cellular damage early in the pathway when the disability is not there and the resilience that is residual in the individual is still strong, and intervention are likely to be much more effective. Thank you very much for your attention.
Thank you very much, Dr. Ferrucci. Now we are going to our second speaker, Dr. Howlett.
Okay, so I'm hoping I'm sharing my screen.
Yes, we can see it.
Perfect, and I'll get a pointer. Hi, well, thank you so much for inviting me to participate in this seminar series. And all I can say, Christiaan, is I'm glad I'm not going last. That was a great introduction and tour de force, Dr. Ferrucci. So I'm going to try to, take up some of the same themes, maybe with more of a focus on some of the work in animals that we've been doing. And I do tweet occasionally, and this is my Twitter handle in case anyone's interested. So we know well, this group knows well, that in 2001, although there's many frailty tools that are used now, there's two approaches to operationalize frailty that have really been widely used clinically. These include the frailty phenotype, where frailty reflects deficits in 5 physical domains, and the frailty index, which counts deficits in health in large numbers of different health domains, usually more than 30 items. And notably, both of these approaches, the phenotype and the index, have been reverse translated from people to be used in mouse models and in other animals as well now. I'm not going to talk about the frailty phenotype, happy to do that in discussion. My work has mostly used this tool to look at frailty in mouse models. And we developed an instrument in 2014 where we looked at different bodily systems in mice, and we looked at what normally happens as mice age. And we looked at health deficits, and we looked at the accumulation of changes in these maladaptive aging changes in the mouse, in terms of their behavior, how we did the physical exam, et cetera. So we do kind of a mini comprehensive geriatric assessment of the mice. And much as is done in people, we get a frailty index by dividing the deficits that we see in these different domains in an individual mouse by the total number, and there's 31 deficits in this list. And then we get a score between 0 and 1, with higher numbers denoting greater levels of frailty. And we've published extensively on this. This was just from our first paper, but the frailty index score we found when we compared a group of data in humans with our data in mice, when we normalized age as a % of maximum lifespan, we saw that really frailty index scores increased with age in both mice and in humans. And in subsequent work in this paper in Scientific Reports in 2017, we did a head-to-head comparison with human data, if anyone's interested. And we have a recent Nature Aging review that we published in 2021 that really published with Andrew Rutenberg and Ken Rockwood, where we've actually looked at frailty index from the mice work and also the human work, if you're interested. So in terms of frailty mechanisms, well, there have been hallmarks and pillars of aging. This is from Brian Kennedy's paper in Cell, he's recently published another one in 2022, looking at pillars of aging, and I would argue also frailty. And they include things like macromolecular damage, so DNA damage, changes in the epigenome, dysregulated responses to stress, changes in proteostasis, protein aggregates formed, dysregulation of mitochondria leading to metabolic dysregulation, exhaustion of stem cells, and also chronic inflammation. And so work in my lab has started to do a little bit of looking at chronic inflammation and its role in frailty in mouse models. So what we've done is we've simultaneously measured a number of different cytokines from small blood samples from aging mice. And we used serum, and we measured frailty with the frailty index tool. We used mice that are between 21 and 23 months of age in this study. And that would be roughly equivalent to 65, 70-year-old human years. You could argue about that, but it's just to give a frame of reference. And we did publish this work in 2019, but what we saw was various pro-inflammatory cytokines, importantly here IL-6 and IL-12P40. And we measured, as I said, a lot of these cytokines in this panel. But we found a number of these pro-inflammatory cytokines increased as the frailty index score increased. And so you see that we see no significant effect here for IL-6 in males and IL-12P40 for females, but we saw that different cytokines, pro-inflammatory cytokines, correlated with the degree of frailty, and that there was a sex-specific effect, so that they differed between males and females. So we tried an intervention over the long term designed hopefully to reduce inflammation. We actually used an angiotensin converting enzyme inhibitor. We used enalapril. And what we did was we treated mice from about age 16 months, in the case of the males, all the way up to age 26 months. And what you can see here, this is the frailty score versus age, older females, older males. You can see the enalapril treated group had really lower frailty scores than the control group. And in fact, giving them enalapril appeared to arrest the increase in frailty in the females. And similarly, in the males, we saw a decrease. It was arguably a smaller effect, but we did see that frailty also declined in males. So with respect to inflammation, we wanted to look and see whether or not the effects were mediated, at least in part, through impacts on inflammation. So it's kind of a busy slide. It shows a number of the different cytokines, chemokines that we looked at. And we saw middle-aged and old mice. These are the females. And the bars that are black are treated with enalapril. And I think what you can appreciate here is in the females, we saw an increase in a lot of these cytokines with age. And when we treated with enalapril, we saw a decline. And in a number of cases, this decline was statistically significant. So suggesting to us that some of the benefit that we were seeing from this enalapril treatment might be due to changes in the degree of chronic inflammation in these mice. Now, we didn't see the same pattern in males. We saw some cytokines that did increase, like IL-6 with age in males, but when we treated with enalapril, we actually found that the enalapril increased IL-10, which is an anti-inflammatory cytokine. So again, this is a really interesting difference, suggesting that the relationship between inflammation and frailty may be different in males and females. More recently, we've looked at a different intervention, which is to look at an exercise intervention. So we had old mice, and we allowed them to either be sedentary or to access an exercise wheel, which we quantified the amount of exercise they did. And what we found was we started these mice and did about a 13-week intervention. And what you can see here, in frailty index plotted against time, this is end point, we found that frailty index scores actually declined with age in females when they were allowed to exercise. And they declined, they didn't go up with males, they didn't actually decline. But again, we saw a very strong effect in females, and we also saw a beneficial effect in males. So I guess the next thing is, what relationship did that have with inflammation? And we found there was no effect on serum cytokines in the males. So the bars are exercise and the open are sedentary. You could argue there's some small signal here. And in the females, there was actually some tendency for some cytokines to actually increase with exercise. So we feel as though the exercise may be benefiting a different mechanism of aging and frailty than the enalapril. And that would lead us to think about maybe testing multiple interventions in the same population. So we go to the Geroscience hypothesis, where we have all of these subcellular and cellular mechanisms of aging and frailty that start to accrue damage with age. And then this goes on to affect organ and function and system failure. So if we treat fundamental mechanisms of aging and frailty with interventions like exercise, like some geroprotectors such as enalapril potentially, we can theoretically slow the decline in organ function, delay the onset of disease, and hopefully improve lifespan and health span. And so that's the type of thing in my lab that we're trying to pursue, would be like a multimodal intervention in mice to see if we can target multiple mechanisms at the same time. So in conclusion, we found that an enalapril treatment reduces frailty, at least in part by effects on chronic inflammation in mice, and this differs between the sexes. Late life exercise is also effective, but it doesn't appear to be related to impacts on inflammation. Sex differences are apparent, and even the mechanisms may be different. And finally, I think this is an exciting idea that multifactorial interventions, like a lifestyle intervention such as dietary or exercise, plus a geroprotector of some sort, could target multiple frailty mechanisms, and they might be appealing to targets frailty. And I think they can be tested quite quickly and informatively in mouse models. And so we hope to be doing that in my lab. And just people who've helped me with this, lots and lots of them. And I'll stop now in the interest of time. Thank you.
Thank you very much, Dr. Howlett. That was fantastic. And now we go to our third speaker. And just a reminder to everyone to submit your questions via the questions and answer function Zoom. And we will have enough time at the end to have an interactive session. Christiaan?
Am I showing this screen, full screen this time?
Thank you.
Yeah, sorry, Luigi. So yeah, I'll talk a little bit about, a specific area, and dear to my heart. It's iron homeostasis and the role of biological aging. We know iron is essential for life, and we know it's important for oxygen transport. You can see in the little inset on the right, iron binding to oxygen. So there's this special relationship between iron and oxygen that probably has evolved over time. So besides transport of oxygen, iron is also important in the mitochondria. So the oxygen gets eventually to the mitochondria, your organelle that produces energy. And you can see that iron is incorporated into the complexes, of the mitochondrial inner membrane that help generate a proton pump in the form of iron sulfur clusters. Also, iron is important in the TCA cycle as a conatase. And, this is, the major cycle to produce reducing equivalents. So very important. Also, you can see in this slide that there are other metals like copper and zinc that are important in energy production. And they do have something similar that some of the transporters we're talking about today. They transport iron into the cells, but they also transport copper and zinc. So let's not forget about those, but we will not discuss those in extent. Also important is that iron is involved in many other, sometimes unknown mechanisms of catabolism and anabolic processes. And I'll give you one example of that later in the talk when we talk about muscle wasting. And it's so important for immune function. Now I got to go to the next slide. So here is a little bit of the conundrum of iron. Iron is essential. We need it. We get 10 to 20 milligrams in our diet, but only very careful amounts are absorbed every day, 1 to 2 milligrams a day, because iron is essential, but also toxic. We lose 1 to 2 milligrams a day. So we don't want to accumulate iron in the body, in cells over time. So we have absorption, a couple milligrams, and then removal. It's transported by transferrin. Of course, most of it goes to the bone marrow and generates red blood cells. And some is stored in tissues, ferritin, in the liver, the heart, and we'll talk about muscle. And then some is needed for other processes. So excretion occurs at a steady rate through turnover of intestinal epithelial, blood loss, and also skin that dies off. So absorption is mainly through the gut, but both are affected with aging. We already learned from the first slide that there's different forms. We have heme iron, for example, very important, and in electron transport, carrying, and there's also non-heme iron. Non-heme iron is like storage iron, like ferritin, for example. This is what's happening as we get older. This storage form of iron, like ferritin, there are other forms as well I'll talk about, but they seem to accumulate in a variety of tissues, and they're strongly associated with disease. Causality is always more challenging, but that's one thing we want to investigate and study more. But there's accumulation of this, especially non-heme iron, in a variety of diseases you can see here depicted in this slide. So what might drive this imbalance? To say that inflammation is maybe the major cause is probably not correct, but mechanistically so far, they have shown that IL-6 can affect a hormone called hepcidin. Hepcidin is the master regulatory hormone of iron in the systemic circulation, but also important in removing iron from the cells because it regulates a protein called ferroportin. Ferroportin is located in the gut, and also on a variety of cells to help export out of the cell when needed. So this hormone you'll see later is in imbalance with old age. I'll show you that. But this master hormone, again, is very important to regulate the amount of iron. So we have a double whammy occurring as we get older. And I'll show you some of that data. At the time we have iron accumulation, like ferritin, and this can cause oxidative damage and a variety of disease conditions. On the other end, we have an issue of iron deficiency often occurring, where issues with iron absorption at the gut are affected potentially by inflammation. Here again, just to make it clear, the different iron forms at the top, you have heme iron, hemoglobin, myoglobin. Those are very important forms where iron is needed. At the bottom, non-heme iron, of course, is important as well, but the issue becomes that we have excessive storage of ferritin in cells, and this is associated also with an increase in labile iron. And this is where the problem becomes very toxic and dangerous, because this labile iron is redox active and can cause mutations, deletions through phantom chemistry, where free iron cleaves peroxides in the cells and makes hydroxyl radicals and alters DNA and causes strand breaks. So besides ferritin, there's other forms of storage iron that increases in the cells. Probably the most famous or infamous one is maybe lipofuscin, which is an undegradable form of lipids and iron and metals and whatnot that accumulates, and again it increases the risk for the amounts of labile iron in the cells that can cause damage. Okay, back to hepcidin. We have shown, and others have shown, that with aging, hepcidin levels are increased. Again, this is this hormone that regulates the amount of iron that's absorbed in the gut, but also regulates iron that's can be removed from tissues. You can see in young subjects around the age of 30, this is muscle tissue, this is plasma, sorry. We'll talk about muscle in a second. If you compare them with high functioning, so relatively resilient people that are high functioning based on a SPPB score, a short physical performance battery score, there's a slight increase. But if you see low functioning individuals, the amount of hepcidin is significantly greater. So it's hinting towards an increase of hepcidin, and then therefore a breakdown of this important protein, ferroportin. What happens to iron import is also critically important. We need useful, fresh iron import, so to speak, from the cell, and this is mainly done by the transferrin-bound iron transport mechanism. It's done by the transferrin receptor, and iron is imported into cells because we need this to go, mainly to the mitochondria for all those important functions that occur there. And this is what we're dealing with as we get older. There is a significant drop in the transferrin receptor. I mean, I've been involved with studies in aging. I've hardly ever seen this large of a drop, especially in these low functioning individuals. There's very little transferrin receptor to bring iron, useful iron into the cell that can be transported. At the same time, in these studies, we find increases in ferritin in this storage protein. CYP8 and CYP14 show a similar trend. These are receptors that are typically known to import zinc and copper, but they also import some iron. But you can see the major import mechanism of iron is impeded in skeletal muscle of very low functioning older individuals. Okay, why is it not moving? There it is. This is an example to show you how important this receptor is. This is just published this year. It is in a very different condition. I am aware of this. This is cancer cachexia. This is a condition where there's heightened levels of inflammation and a massive amount of muscle loss very quickly. But it just shows you that in this paper, they did this paper in both animals and humans. They found a massive downregulation of this transferrin receptor and also at the same time an increase in ferritin. So very similar to what happens with aging. You can see their model here on the left, and they found also muscle atrophy in these animals and in humans. And then they did a simple intervention of iron supplementation, and they were able to preserve, increase mitochondrial function and preserve muscle mass in the animal models. Even in the small human study they did in this paper, they were able to show that iron supplementation, increases grip strength in these individuals. So again, it's very important to have functional transferrin receptor in muscle. Maybe due to time, I won't talk too much about it, but there's also deregulation of import mitoferrin or compensation mechanisms of increased mitoferrin to import iron into the mitochondria. And there's a massive decline in frataxin. Frataxin is very important in distributing iron in the mitochondria. And you can see, for example, frataxin levels are massively declined in the muscle of these very old 80-year-old individuals. And we certainly know that frataxin deficiency with Friedreich's ataxia, which is a genetic disease, causes difficulty walking and neuronal issues and loss of sensation in the arms. So in summary, tissue non-heme iron and labile iron is increased at old age. Mechanisms of iron uptake in, for example, skeletal muscle transferrin is impeded. Export ferroportin, possibly regulated by or caused by the increases in hepcidin, and there's an increase in storage of iron levels by ferritin. So there's a deranged amount of iron in especially the older adults. I didn't show the paper, but we also published a paper where the mitochondria that have a lot of non-heme iron, they're also more susceptible to ferroptosis or to permeability transition pore opening. So longitudinal studies will need to investigate the role of metals, not only iron, but also copper and zinc, I explained, on mitochondrial function, ferroptosis, and functional and physical decline. I want to thank investigators at the University of Florida, Northwestern University, and Luigi Ferrucci at the NIH.
Thank you very much, Dr. Leeuwenburgh, for this fantastic presentation. I really want to thank the three speakers for the fantastic management and your time, and also with the connection with the clinical implications. So that I think that's very useful for this panel. So we have got a lot of interesting questions, but I will start with one question per speaker, and then we will start, an open discussion. The first question that I got here is for Dr. Ferrucci. Regarding you said in your presentation that there is the biological manifestations go first. But the question that I have here that I receive is what triggers those biological manifestations? So still the biological changes are manifestations, but what would be the upstream mechanisms that trigger that, and are those mechanisms shared?
Well, that's a great mechanism, and you know, I think that what's really happening is that entropy is what determining, you know, those damages. You know, each molecule exists in a stable state, but there is accumulation of damage, you know, both because of environmental stress, but also because some of the repair mechanisms tend to fade and not to be so efficient. And of course, you know, you can have, you know, a lot of damage accumulating in the cell because of redundancy. This may not be seen at the level of any phenotype. But that can be detectable if you have this special tool that allow you to do that. And then if we could detect that and address that at an early stage, we could, you know, prevent the following consequence at the time, whether the organism is still able to react, because many of the other resilience strategies are still, you know, there and are still functioning. What we do, unfortunately, is that we only intervene when a disease occurs. And when the disease occurs, you know, most of the things that are cause the disease has happened, and the resilience strategy that we're trying to fix the problem and avoid and repair the damage is no longer available. And so our intervention are less effective. And I think that's exactly why this is the mechanism that is occurring. It's true at every level. I'm giving you an example, but I can give you others. For example, you know, Christiaan was talking about mitochondria. You know, mitochondria are healthy if their quality control is well functioning. At some point when the quality control no longer work, you know, these you know, fragmented and dysfunctional mitochondria stay in the cell, and we know that the mitochondria is one of the center, you know, of the inflammatory response. And so defective mitophagy end up being a cause of inflammation because of mitochondrial dysfunction. Again, if this is limited to few mitochondria, they have hundreds of mitochondria in the cell, nothing is going to happen. But it's become a generalized phenomenon, then it start having functional consequences.
Thank you. Any comments from the other two speakers, on our speakers, Susan or Christiaan?
You know, I guess just to elaborate on that, we've just published a paper in eLife where we actually worked with Andrew Rutenberg's group in physics here at Dalhousie and looked at rates of damage and repair in both mouse and human data. And so we find that there are actually changes in the both the ability to repair damage and also the ability to resist damage, right? So the two are changing in the same mice or the same people, and so we've modeled that with that group. And so, you know, I think that's what's happening. You know, you're getting two things that are going on. You're not able to resist the damage, and you're not able to repair the damage. And you're looking for maybe in interventions the idea that you can then tweak things so that, you know, you can either repair damage more effectively or resist it. So I don't know if that helps, but
yeah, no, I agree. And you know, another example is maybe immunosenescence, where you know your immune system is not very functional. It's important for, you know, a lot of things we know. And so if there's no early detection, you know, the resiliency of that system goes down. It can affect muscle, it can affect liver, it can affect the heart. So it has multiple effects, you know, even on repair. So it's they're all working together, and they're, you know, they're weak links, you know, showing up differently in different people. I mean, look at the mouse models that you're investigating. They start all pretty much the same litters, but there are some differences as they get older, which is very interesting.
Excellent. Okay, thank you very much. We move to another question, and this question came from Ariela or Kavi that's addressed to Dr. Howlett. is it possible that inflammation is a bystander and not on the causal pathway of frailty?
Yeah, for sure. You know, and it could be, you know, that it comes about secondary to other damage, right? So it would be hard to, that would be a hard thing to show for sure. All I can say, and I didn't really mean to imply that the inflammation was causing it, but, you know, I think that there's definitely a link between inflammation and frailty, and they're definitely correlated in our mouse studies, right? So, you know, I think that provides some evidence for, you know, quite a nice link between the two of them. But we saw no such link in the exercise work. So, in fact, if anything, in the females, we saw the opposite, whereby higher levels of inflammation were correlated with better, you know, better aerobic performance, if you will. The paper goes into more detail there. So obviously, that's not the whole story, right? And I think that that's why we're really interested in some of these multifactorial interventions. So you could maybe get at a couple of different pathways that are implicated in aging. And hopefully, you know, what we do know is the enalapril treatment did reduce their levels of frailty. We also know that for the exercise. So I hope that answers your question, but I didn't really mean to imply that it was the only thing. It's what we've looked at so far.
And thank you. And actually, that question links with another question from Jeffy Ben to Dr. Leeuwenburgh. Can inflammation cause functional iron deficiency?
Yeah, so in the extreme study I showed with cancer cachexia, again, you know, sure, cancer cachexia is multiple things go wrong, but one is there's heightened levels of inflammation. And, you know, the receptor that went down in that study is transferrin receptor. And at the same time, you know, ferritin goes up, which, you know, is a storage protein of iron in the cells. And a simple supplement of iron was able to overcome some of this, of the deficiencies, and, you know, muscles were getting bigger again, the mitochondria were working again. So yeah, it's, that's a definitely, you know, causal study. It's probably more subtle with aging and muscle, but yeah, look at the drop of transferrin receptor in those muscles of those very, very old people. That was quite stark. And whether that's only caused by inflammation, I don't know. I think it's multi-complex.
Can I make a comment here? I think
Yeah, sure.
that you know, it's white and black at the same time. You know, we need the inflammatory response, because the inflammatory response is a positive response. Inflammation is a good thing. I think that the point is when inflammation overcome, you know, what are the needs of the individual and become, you know, a problem. You know, two things. One is inflammation is caused by stress. The stress response, for the first thing that the stress response does is block protein synthesis. And in the acute term, blocking the protein synthesis is a good thing because you can allow the proteostatic response to function. Over the long time, if you don't synthesize protein, you get problems because you cannot renew the tissue. The second thing I want to say is that the fact that inflammation is a little bit also causal, I think it's been demonstrated in the CANTOS trial. The CANTOS, that is the block of IL-1 beta, you know, prevented cardiovascular event very, very strongly. But very few people know that there is a number of, you know, post-analysis of the database showing that it also prevented lung cancer, it also improved diabetes, it also reduced hospitalization for congestive heart failure. So it's not only cardiovascular disease, but it really affects the entire physiology of the individual. So I think that targeting inflammation, if we could find the right way to do it, will still be very valuable.
Yeah, I agree.
Sorry, go ahead.
Yeah, and senescence again is another example. Those cells produce a heightened levels of cytokines and chemokines, and if they're knocked out, those animals do live longer. And you know, there's a lot of clinical trials now that are targeting those senescent cells. So it's probably not going to be the only thing and the holy grail, but it will be something that will happen there.
Excellent. And Dr. Ferrucci, I have a question for you here from Bruno Cavalcante. He says, actually, really glad to hear you, big fan. How can you explain that some individuals that are living in environmental marked by social inequities, such as poverty, low access to health sources, violence, and so on, and even exposed by such environment, they present certain resistance to develop chronic disease? Any comments on that?
I think that you're talking about a small percentage. I would say that in general, the people that live in environmentally unsafe and low socioeconomic status, they tend to have health disadvantages. But you're right that there is evidence of a small number of people that seem to become incredibly resistant. And I think that we always think about a linear relationship between stress and response, but in reality, we know that there is something that's called hormesis, is that in order for us to, empower our defenses, we need a constant small stimulation. So if an individual have a very strong resilience response, and so can consider the continuous stimulation as a normatic mechanism, maybe a few individuals use that continuous stimulation to develop a strong resistance. And in fact, we find some supercentenarians in place where are the least expected. And I think that that's where, the proof is. Of course, we don't have, scientific proof that this is true, but I think that some of the evidence coming from the supercentenarian study from the Keio University in Japan, for example, suggests that the accumulation of senescent cells in this individual is less than in any other individual, even that are 20 years younger. So there may be some, resilience mechanism at place there.
Thank you. I just compiled a few questions to Dr. Howlett because people are very interested on the first choice of enalapril, because people, Namrata and Blanca here mentioned that they don't typically think of it as an anti-inflammatory. And some people also are interested on the responsiveness of females versus male, so underlying biological reasons. So there are two combined questions there.
Great. Yeah, I can try to answer all those questions when we're done too, if we don't get to them, but I saw those as well. So first, with respect to enalapril, I agree it's not what you would normally think about as being an anti-inflammatory. So we did choose it, a couple of reasons for that. One of them was there was some interesting papers by other people showing that you could actually administer enalapril in these higher doses. I think Christy Carter's lab might have done it in rats. Anyway, we sort of looked at that and looked at the impacts it was having on overall function, and they hadn't measured frailty, but we thought it might be a good frailty intervention. There's also some work that shows that, for example, mice with an angiotensin 2 type 1 receptor knockout actually live much longer than wild type mice. So we thought that it was interesting because it had this impact on longevity, given that if you knock out the receptor, you get an extension of lifespan in rodent models. So we thought that the other thing is, I'm a cardiovascular researcher, by nature, although I've sort of expanded into other systems, but, we were looking for some cardiac remodeling as well. So for those reasons, we sort of chose enalapril. And we know that angiotensin 2 promotes inflammation. It promotes fibrosis in different tissues, including the heart. And so we were interested to look at whether or not enalapril we hypothesized it would be beneficial in frailty, and we did find that it was, right? So now we need to figure out more about the reasons. Someone asked what the dose. We got the dose from another study. We didn't do a dose response curve. We did an initial study. We actually even found an impact in animals that were middle-aged, so between 9 and 12 months. Only in the females, we saw a much more profound response. So in that same paper, we piloted the work in a younger group, and then we worked in an older group. So the females seem to be more responsive. And one reason for that could be, and this would be the case in exercise as well, but you can really see it in the data on the enalapril study. We found the females were frailer than the males, and we've actually reported on that. And that kind of dovetails with clinical observations. So it suggests some really interesting biological underpinnings for the idea that women have higher frailty scores than men. And interestingly, they tend to live longer. We don't necessarily see that longevity piece in the most studies. They're more difficult because we have animal husbandry rules and humane endpoints, et cetera. So it's more difficult to know the answer to that. But for sure, we have seen the females frailer than the males. So maybe the effect is more impactful because there's a larger signal and that's being reduced by treatment. I don't know. And we're trying to look at whether or not there are other reasons for it around differences in the immune system, for example, and other differences that could be there between the male and female mice. I think I answered those questions.
Yeah, excellent. Thank you. So I think that because we are over time now, I would like to thank the three panelists. This has been a fantastic seminar. We had tons of people in the room, more than 220 people attended. And this is recorded. So as I mentioned, you will have access. So you want to share the links with your colleagues. We are more than happy to share. The Global Frailty Network is growing, and we expect that, or we hope that you are finding these sessions very interesting, and also and that you will join our initiative. So with this, I just want to invite you to our next webinar next month by Professor Leocadio Rodriguez-Mañas from Getafe Hospital in Madrid. And Leo is going to talk about frailty during and after hospitalization. So I hope we will see you again next month. And thank you very much for joining us. Thank to the speakers again, and have a nice afternoon, everyone. Thank you.